Okay, let's learn a little bit more about how digestion of proteins takes place in particular. Now, to be very comfortable with the digestion of proteins, we need to be also very comfortable with the structure of proteins. Okay, so I'll do that a little bit here. Proteins are large molecules and they are insoluble. And we need to break them down into smaller soluble molecules so that they can be absorbed. Okay, this large insoluble molecule can be called a polypeptide or a monomer, polymer, sorry, and this would be our monomer. The amino acid, that is the monomer, has this structure here, so make sure we're just referring back to biological molecules when we think about digestion of proteins, and what we're making sure happens with our protease, our collection of protease enzymes, many of them. We're making sure that we are breaking these bonds to separate our amino acids. We're breaking the peptide bonds. Okay, that's the whole point. So we've got a bit of a recap here on the structure of the digestive system. Okay, taking food into, and we've got some very concise bullet points here that tell us step by step just what happens to the proteins. Let's not worry about anything else. This is just proteins. So this is the collective work of the different proteases at different parts of the digestive system. Okay, first thing that's going to happen in your mouth, you've got mechanical digestion, breaking down your large chunks of food into smaller ones. Once the food gets into the stomach, we have our hydrochloric acid to kill the pathogens, and we have an enzyme, a pepsin. Now, pepsin is a type of protease. This pepsin works at low pH because remember, it's in the stomach where stomach acid is being secreted. Okay, this pepsin only works in the stomach. It can't work anywhere else. It is an enzyme where the optimum pH is low. So pepsin, optimum pH would be like this. That would be pepsin. Okay, optimum pH pretty low. We get further digestion, hydrolyzing these peptide bonds. Okay, and we get a bit of mechanical digestion in the stomach as well because the stomach churns the food, the wall moves to mix the food. That's considered to be mechanical. Let's get to the pancreas now, over here. Pancreas secretes another protease enzyme called trypsin. But this time, this trypsin being released by the pancreas is going to work on the food here. But the food here, or the digested food here, has already received bile from the liver to emulsify the fats. And the bile does two things. It emulsifies the fats, but it also makes the pH of the digested food okay for us. We don't want low pH coming out of the stomach with the food. We need to neutralize that quite quickly. Okay, because that stomach acid is going to be mixed with the food and we don't want that to carry on through the digestive system. It would be damaging. So we need to neutralize, we need to change the pH of the food as well as start to break down the bile. And that's what trypsin does. Okay, oh no, that's what bile does. But trypsin, when it is released, now this trypsin is going to work not at a low pH but a high pH because now everything's gone a little bit more alkali or slightly neutral, slightly alkali because of the action of the gall or the bile in the gallbladder. So for trypsin to work, we need to get a different enzyme to turn inactive trypsin into active trypsin. And that is the work of this enzyme called enterokinase. It takes trypsinogen, which isn't active, and turns it into trypsin, which is active, and then it can go do its work. Okay, then what trypsin does, as we've said before, it breaks down the peptide bonds between the amino acids. All right, so we've got pepsin in the stomach, we've got trypsin in the pancreas, but it's not active. It's got to be activated. It's got to be turned into active form. So that is where Enterokinase comes in. Enterokinase is an enzyme that takes trypsinogen and turns it into active trypsin. Okay, and then the last we've got where absorption takes place. And I'll leave you to make notes on this to support your studies.